DNA fingerprinting. So in the DNA fingerprinting is like normal like our fingerprinting only with the use of analyzing the DNA sequence. So normally in DNA the bases 99.9% bases are similar in all individual of a species. But in every person the 0.1% DNA bases are different from one person to another person. So with the use of these differences only we conclude the which person the person or the DNA இது. So அதை conclude பண்ணலாம். That the particular technique is known as DNA fingerprinting. So DNA fingerprinting la repetitive DNA are present. நான் லாஸ்ட்ல சொன்னல human genome project. The adenine thymine cytosine guanine adenine thymine cytosine guanine in some millions of bases க்கு அப்புறம் ஒரு particular sequence are repeated in n number of time in individual to individual. இப்போ எனக்கு ஒரு டைம் ரிப்பீட் ஆயிருக்குன்னு வச்சுக்கலாம். For example adenine thymine adenine thymine adenine thymine ஒரு 10 times ரிப்பீட் ஆயிருக்குன்னா but in another one person same cytosine guanine cytosine guanine cytosine guanine 10 times ரிப்பீட் ஆயிருக்கும். So in person to person in a species some DNA bases are repeated n number of times. The repeated bases are repetitive DNA. It is actually useful for the DNA fingerprinting. DNA fingerprinting ஒரு discipline ஒரு branch develop ஆச்சு அப்படின்னு சொன்னா with the use of repetitive DNA. And the next one is satellite DNA. This satellite DNA is nothing but this is actually a chromosome. Chromosomes la centromere, that is primary construction, and this centromere adukapram pathina idu chromatids, this centromere, and the secondary construction or area varum. Secondary construction, and above the secondary construction the part of the chromosome and the part ku peru satellite chromosome. In satellite chromosome also the DNA are present, that is satellite DNA. The satellite DNA is differ from in person to person. Over person ku satellite In DNA fingerprinting, that unique phenotypic appearance, like எல்லாம் கொஞ்சம் பார்த்துட்டு இருந்தோம். Then the satellite DNA and repetitive DNA. Satellite DNA is nothing but the DNA present inside the satellite chromosome. Satellite chromosome na idan. Listen, this is normally a chromosome. And chromosome contains two chromatids and one centromere. In the centromere, that is primary construction. So adala centromere irukum. That is secondary construction. Secondary construction also contain one part of the chromosome. So that part is known as satellite chromosome. In satellite chromosome, the DNA are present. The DNA is nothing but satellite DNA. In satellite DNA, in every person, individual. For example, one person satellite DNA in adenine thymine like order means in another one person satellite DNA is different. Suppose cytosine guanine. In another one person guanine cytosine. In another one person adenine thymine cytosine guanine adenine thymine. So in each person in a population, each individual in a population, different types of satellite DNA are present. With the use of these satellite DNA only, we conclude the DNA and DNA fingerprinting technology is developed. So the first developed with the use of Alex Jeffries. Our the first start pannaru. So satellite DNA oda use vechi dhaan actually we are planning to identify the thief, culprits. Various research எல்லாம் பண்ணிட்டு இருக்காங்க. அதாவது the satellite DNA only is important for the DNA fingerprinting. Okay, so the next one, this satellite is divided into some categories like micro satellites, mini satellites, micro satellites and mini satellites எல்லாம் முடிச்சிருக்கு. Then even human, all the cells contain DNA, we know. Then RBC, WBC, RBC may be enucleated in matured state. WBC contain nucleus and platelets also contain nucleus, and as hair follicle, the base of the hair, the starting point of the hair also, this contain DNA cells, and the skin contains cells, and bone contains cells, saliva and sperm also contain some cells and DNA. So for example, ஒரு person இன்னொரு பர்சனை மர்டர் பண்ணிட்டாங்க. The murdered person, ஒரு 5 persons suspect பண்றாங்க. So the first scene, the crime scene placeல check பண்ணும்போது small part of hairs are present. The hair is actually contains some hair follicle. Hair follicle contains some cells. Then the hair follicle cells is different from the dead person. Appa some new person is arrived in the particular scene. So appa the new person yaar. So the first we take the investigatory department, that is a forensic science department, taking the hair and analyzing the DNA in the automatic sequence machine, analyzing the DNA strands like in the base pairs. And if for example 5 persons are suspected, means the 5 persons on the DNA with the use of blood sample and the DNA eduthu compare the DNA with the hair follicle DNA. So and the person related to the particular crime scene. So easily the forensic science department and the CBI and other CBSE police and particularly crime science, criminal science, criminology and the scientists easily identify the particular person which yaar murder pannadhu appdin easily identified. So with the use of DNA fingerprinting only this is possible. Okay, so same like our bone. For example, ஒரு person ஒரு murder பண்ணிட்டு அந்த person bury இது பண்ணிடுறாங்க, burn பண்ணிடுறாங்க. So only a small part of the bone is extinct. The forensic science department collecting the bone and analyzing the DNA in the bone, and afterwards we conclude this person dead on a person ivrudha with the use of parent DNA. For example, ஒரு அந்த nearby area la yaar and the person oda and the particular child is missing அப்படினு சொல்றாங்களோ அந்த parent oda DNA plus and the blood bones la irundhu edutha DNA. So analyzing the two types of DNA. So lastly, the forensic science department concludes, so the person is in the இவங்களோட child or இவங்களோட parent. So with the use of DNA fingerprinting only, with the use of saliva also contains some epithelial cells, squamous epithelial cells, and the cells analyze panna the person saliva yarodadu appdin is identified. And sperm contain DNA. So a rape scene nadandirukku. With the use of sperm, we analyzing the sperm content and which person is raping. So raped. So on the particular crime scenes, it is DNA fingerprinting and useful. With the use of DNA fingerprinting, even paternal disputes. For example, one child is ரெண்டு பேரண்ட்ஸ் இருக்காங்க, the child argue பண்றாங்க, this is my child அப்படின்னு சொல்லி. With the use of DNA test, with the use of DNA fingerprinting test, automatically the doctors knows, so when the child is belongs to which parent. Okay, so these like DNA fingerprinting, it is a various applications in our day-to-day life. So inheritable mutations, DNA polymorphism. For example, with the use of DNA fingerprinting, we analyze the evolution also. With the use of molecular biology technique. Molecular biology means what? Molecular phylogeny means what? With the use of DNA analyzing, DNA base pairing, we conclude that the evolution taking place in a disorder. So evolution taking place in a disorder அப்படினு சொன்னோம்னா with the use of molecular phylogeny identifies. So evolution also with the use of DNA fingerprinting and molecular phylogeny, DNA வை compare பண்றது. For example, human DNA compared with or 5 variety of monkey DNA. So which DNA is similar, more similar or most similar to the human DNA அப்படினு சொன்னோம்னா the human is developed from the particular monkey during evolution. So evolution points of view, molecular phylogeny, various crime scenes identification, the DNA fingerprinting technology is very useful. Okay, yes. DNA fingerprinting technology first are identified or used by Alex Jeffries only the DNA fingerprinting technology first legal use பண்ண ஆரம்பிச்சாங்க. Okay, then what are the steps in DNA fingerprinting? How we analyze the DNA structure or the DNA fingerprinting with the use of various methods? What are the steps are involved? Variable number of tandem repeat is the basic, because we know that the chromosomes contain a satellite chromosome. In the satellite chromosome, some DNA segments or DNA base pairs are repeated n number of times. நம்மளுக்கு தெரியும். For example, ஒரு person அப்படின்னு சொன்னோம்னா அவனோட base pairsல ஒரு particular base pairs is repeated more times. In another one person, another one base pairs only repeated different times. So that the particular character is important for the DNA fingerprinting. The characters are VNTR, variable number of tandem repeat. Variable time, the DNA base pairs are repeated in the total DNA content of a particular individual. But in another individual, different types of base pairs are repeated in n number of times. In another one individual, another one time repeated. So with the use of VNTR only, they are able to identify the structures. The next one, what are the steps in the DNA fingerprinting? First, isolation of DNA. Isolation of DNA எப்படி பண்ணணும்? For example, in our 12th standard practical also, in a major experiment, isolation of DNA are present. For isolating the DNA, simple steps only. You take a plant material or maybe animal material or maybe forensic science material, blood or maybe saliva or maybe anything. So first we take a simple, I give a small isolation of DNA, simple isolation of DNA. We take a small piece of fruits, this is for example banana fruits, and add a small amount of detergent solution. Detergent solution means soap oil. So full nalla mix பண்ணனும் இந்த banana plus detergent solution, nalla mix பண்ணிட்டு thoroughly mix the solution, Then they filter the solution, banana extract with the soap solution, அது கொஞ்சம் filter பண்ணிக்கணும். Then the filtered solution add ethanol, methyl spirit, chilled ethanol, not normal ethanol, நல்ல chill பண்ணி ethanol add பண்ணினா within ஒரு 5 to 10 minutes, so DNA slowly isolated, slowly separated from the pool and it is மேல் அப்படியே float ஆயிட்டு இருக்கும். So this DNA. So with the use of simple method we collecting the DNA in a plant sample, but in blood sample, in a blood in the bone sample, from the saliva means it is a complicated technique. So normally we isolate the DNA from the plant sample means it is easy, but here in the DNA fingerprinting, so we normally isolate the DNA from hair follicles, isolate the DNA from the sperm, isolate the DNA from the blood cells. So the process is complicated, but in day to day life is easily we identify or we isolate the DNA from the plant and animal material also. Okay, so first process in the DNA fingerprinting, isolation of DNA. So DNA isolate பண்ணி எடுத்தாச்சு. Then the digestion of DNA by restriction endonuclease. Restriction endonuclease means is actually enzyme. Restriction endonuclease is the enzyme which cut the DNA in a specific part. So actually these enzymes like restriction endonuclease la in the biotechnology next unit la paakalam. Then restriction endonuclease is nothing but that is a enzyme which is used to cut the DNA in a specific point. For example, EcoR1. So an example for restriction endonuclease. So restriction endonuclease is nothing but that is a enzyme which it cuts the DNA. Okay, so with the use of restriction endonuclease, actually we isolate the DNA, the total length of DNA. Normally we know in the earlier part of this lesson, the human cell DNA is 2.2 meter length in a single cell nucleus DNA, and the 2.2 meter nucleus with the use of restriction endonuclease we cut the fragments, we cut into some fragments. Okay, the digestion of restriction endonuclease. Then the small fragments vanducha, and separation of DNA fragments by electrophoresis. Electrophoresis actually or machine, with the use of electrophoresis machine we separating the DNA and separating the cutted DNA. So the electrophoresis also in the biotechnology lesson la konjam anga konjam paathukalam. So electrophoresis is nothing but the machine which is used to separate the DNA fragments according to the density, according to the size. Okay, so with the use of electrophoresis machines we cut the DNA and separate the DNA. The first cut the DNA with the use of restriction endonuclease, then separate the DNA fragments, because in the restriction endonuclease is cut the DNA in various parts. So after cutting with the use of agarose gel electrophoresis, maybe gel electrophoresis la nariya machines irukku. Agarose agar 11th standard, what is agar? It is a polymer which is produced from the red algae Gracilaria and Gelidium. It is agar the chemical, the solidifying agent. With the use of agarose gel electrophoresis machine we separating the DNA fragments with the use of positive and negative charge, and then afterwards transferring the separated DNA fragments by synthetic membrane. So and the DNA is separated DNA are present in the actually in the gel in electrophoresis la, and the gel agarose gel la and present aayirukkum DNA. Then the DNA is separated and blotted and transfer the DNA with the use of nitrocellulose or nylon paper. Okay, first DNA எடுத்துக்கிறோம், isolation of DNA. Then with the use of restriction enzymes endonuclease we cut the DNA, then add to the electrophoresis machine. The machine it separates the DNA fragments according to the size. Then from the agarose gel and the gel we separate, transfer the DNA into the nitrocellulose paper or nylon paper. That is the step. Then the hybridization using labeled VNTR probe, and the particular DNA fragments இருக்குல்ல isolate பண்ணி cut பண்ணி separate பண்ணி எடுத்த DNA fragments. With the use of DNA fragments யூஸ் பண்ணி VNTR அப்படிங்கிற ஒரு prop யூஸ் பண்ணி அதை ஜாயின் பண்ணும் அந்த DNA fragments. Then detection of DNA by audio radiography, auto radiography அப்படின்னு சொன்னோம்னா auto radiography அப்படின்னு சொன்னோம்னா with the use of ultraviolet rays we see the DNA fragments with the use of orange color and the colors வச்சு பார்க்கலாம். So these all the techniques again biotechnology lesson again ஒரு time வரும். Then அங்க I explain elaborately. This is a simple process. First isolating the DNA and digestion of DNA with the use of restriction endonuclease and separation of DNA with the use of gel electrophoresis machine. Then the transferring the DNA fragments into the nitrocellulose paper and the video labeled VNTR add பண்ணும் அந்த DNA fragments எல்லாம் join பண்றதுக்கு VNTR prop யூஸ் பண்ணி then detection of DNA, how the DNA is similar to the person. For example, a person is actually detected or affected, or example அந்த person தான் நம்மளுக்கு crime scene ல இருந்து DNA கிடைச்சிருக்கு, ஒரு 5 persons ஓட DNA வை அனலைஸ் பண்றோம். So which one is nearly closely related to the suspected person அப்படின்னு சொன்னோம்னா, for example suspected person A அப்படின்னு வச்சோம்னா with the use of auto radiography which person DNA A B C D E, so for 5 persons suspect பண்றோம். So which person DNA is similar to the A, so and the person only culprit. So for identification of this process we use auto radiography. Is actually a machine with the use of ultraviolet rays you can see the DNA fragments with the use of protective glass or with the use of systems. Modern system la paakalam. Auto radiography is a machine used to detect the DNA or observe the DNA with the use of ultraviolet rays. Okay, that is the last technique. And afterwards we identify the person. Okay, so books la kuduthirukam par page number 123. Take your NCERT book page number 123. Take your NCERT book page number 123. This is the drawing for paternal chromosome, parental chromosome and maternal. Paternal chromosome means, listen the drawing, page number 123. First paternal chromosome and maternal chromosome. Paternal means is nothing but father chromosome, maternal means mother chromosome. Okay, then the chromosome number 7 structure, the box like structure is nothing but gene. The in chromosome number 7 நிறைய genes இருக்கு, and the red color box is nothing but genes. And chromosome number 2 also contains some genes, and chromosome number 16 also contains some genes. So in the first one, for example in the particular drawing la we analyzing the child DNA, compare the child DNA with the parents DNA. So normally we know the paternity disputes varum. So one particular child, இது என்னோட child, இது என்னோட child, two parents are arguing in the particular child. So on the particular paternity test, and the paternity test தான் இங்க மாடலுக்கு கொடுத்திருக்காங்க. So actually paternal chromosome 7 from one parent, paternal chromosome 7 from the other parent in the drawing la paru. And the second one paternal chromosome 2 and 2 from one parent to another parent, and chromosome number 16 and chromosome number 16. So upper two parents is actually here, this two parents is A and B. So upper C is actually a child, A is actually one parent chromosome and B is actually another parent chromosome. So actually C is a child chromosome. So a child chromosome is C. In the actually the box like structure is nothing but the mission is gel electrophoresis. Gel electrophoresis mission. The mission is actually the box like structure on the gel electrophoresis mission. With the use of gel electrophoresis mission we analyze the DNA, compare the DNA with C to A and B. So C and AB. So C is actually a child DNA. DNA fragments are separated. Actually the color actually DNA fragments in separated in a such a way, and A also the DNA fragments are separated in the such a way, and B also is fragments is separated in a such a way. So compare the A, B and C. If C the child, So, the C child is similar to the B parent part. Listen to the drawing. The C, the chromosome number 11 is similar to the B and B leper. C is a chromosome number 8 is actually similar to the B. So, A, C and B and the compare panni the distance between each DNA fragments in C and the distance between each DNA fragments in B is similar. So, actually the C child is belonging to B parent, is not the A parent. With the use of this technique, we identify the parents' paternity disputes. Okay. So, the amplified repeats are separated by Electrophoresis DNA fingerprinting. Okay. This DNA fingerprinting technology is useful in various branches like forensic science, identifying the culprit. Various branches the DNA fingerprinting are useful. So, a elaborate idea about the Electrophoresis and the restriction endonuclease and the next unit biotechnology lessons. Okay. So far we are completed 2 units. First unit reproduction in organisms and sexual and the asexual and all the human reproduction. The second one chapter 5 and chapter 6. The evolution lesson is deleted for your board exams. So 2 units are completed. Principles are completed 1 unit, that is health and diseases and microbes in human welfare. The next unit only 2 units left out, biotechnology and population and environment unit, ecology and environment unit. So next class la ecology and environment start pannalam. The first only 2 lessons in the environment lesson, organisms and population, biodiversity and conservation. Remaining 2 is deleted for your board exams. So only 4 chapters are remaining, biotechnology 2 chapters and environment lesson 2 chapters. The last environment lesson in the next class. Organism and population 1 chapter and that biodiversity conservation. Okay. So refer your NCERT, read line by line and in text and exercise questions also refer in the next class.